good evening. We are waiting for some more people to join in. In the meantime, you can tell me if I'm audible. You can just uh, give some reaction. Maybe a like would be good. If you can see the slides and if I'm audible. Yeah, perfect. Thank you. Okay, so good evening, everybody. Thank you for joining in. Today is week 2 of our 12-week course on Genome Editing and Engineering in NPTEL, which is taken by Professor Utpal Bora of IIT Guwahati. I am Sanket Mukherjee, the TA for this course. I'm a Prime Minister's Research Fellow from the School of Biological Sciences at IIT Delhi. So, like we have done previous week, we will be discussing in brief of what are the topics that has been covered during this week. And then we will solve some questions, and you may also ask if there are any questions from your end, and we'll solve it as and when we get those questions. So let's come to the main theme of this entire session. I mean, the entire week has been about DNA damage and its repair. So DNA damage could occur due to uh, either replication error. We'll be talking about all of this in details, or ionizing radiations. We'll talk about UV radiation, gamma rays, and stuff. Then it can happen due to oxidative stress. That is mainly, due to the metabolic processes which happen in humans. What happens? there is production of reactive oxygen species like hydrogen peroxide. Then there is superoxide radicals which are produced, which damages DNA. Right? We'll see how that happens. There is UV radiation, of course, and it is pretty well known that UV is damaging to DNA. And then there are some other mutagens as well. So mutagens are chemicals, or these are substances that can damage DNA, right? We'll talk about it. What are the types of DNA damage? So the types of damage could be either insertion or deletion in the DNA sequence. So we know DNA is made up of 4 letters, A, T, C, and G. It is possible that due to the damage, due to any of these reasons which were listed on the left, there is an insertion of a sequence or a deletion of some sequence that could happen. There could be abasic sites. Abasic site means, if you remember the structure of DNA, so it actually contains a ribose sugar, there is a phosphate backbone, and a nitrogen base. So now if this nitrogen base is removed, then we call this kind of a damage as a abasic site, right? But the rest of the DNA backbone remains. So this is one kind of damage. There could be single-stranded DNA breaks. So only, like, DNA is a double helical structure. On one of these strands, there are two strands. In one of these strands, there could be breaks. There could be nicks or breaks. There could be double-stranded DNA breaks as well. Apart from that, there could be DNA adducts. So these are, some extra structure that comes up and joins, the DNA, and it again causes some problem. Now, there could be other thing, like there could be inter or intra strand cross-linking. So if there are like two strands of DNA and one of these strands, so one base is complementary to the other base, that's how it is done. So if you remember about the Chargaff's rule, it forms double bond with T and then G with C. But sometimes within these strands, there could be, from the same strand, there could be a cross-linking which can happen, or there could be, cross-linking between these two strands, but not, parallel to each other, not as base to base level on the same stack, not like that. It could happen on two different, bases. They can cross-link. And these kind of cross-links are DNA damages. It has some implications. We'll study about it in detail. Now, sorry. Really sorry. What are the DNA repair mechanisms? So DNA repair mechanisms, one of them is photoreactivation. We will talk about it. Photoreactivation. Then there is nucleotide excision repair. Also known as NER. There is base excision repair. Also known as BER. There is mismatch repair. So there are various proteins which are involved behind all these repairs. We'll discuss about it, MMR. Then there is double-stranded break repair. These are the major pathways by which DNA is repaired, right? We'll talk about it. So when we say that a DNA is damaged, right? What do we mean when we say that a DNA is damaged? We mean that it is an alteration in the basic chemical or physical structure of the DNA, which could have further implications which could affect the functioning of genes, or it could also affect the functioning of proteins that are encoded by this gene, right? This is the basic idea. Now, talking about again these kind of damages, there could be like single base alterations, more than one bases are altered. What kind of damages are here? So in case of single base alteration, the first type is depurination. So what happens is, in this case, the purine is removed. So as I already told you, that in case of DNA, there is a sugar, that is the ribose sugar, and the phosphate, backbone, and then there is the base. Now, this base, if removed, if it is a purine base, for example, purines are adenine and guanine. So if these purines are removed, it is called depurination. That is one kind of damage. Another could be deamination. So deamination could happen to, let's say, cytosine. So deamination is removal of amine group. So if it happens with cytosine, then cytosine gets converted to uracil, right? Uh, and if it happens with adenine, then it gets converted to hypoxanthine. So how are these damages like important? Because if cytosine is present in the DNA, it will base pair with guanine, right? That is how it happens according to Chargaff's rule. But if cytosine is converted to uracil due to deamination, we will see that this uracil now, which is present in place of cytosine, will now base pair with adenine. So instead of CG, we will see something like UA base pairing. So this can lead to mutations, right? Then there is alkylations of bases. So this is like another one. 3 is adenine to hypoxanthine. This is also deamination reaction. Then fourth one is alkylation of bases. So if the base is alkyl group is added to the base, then we call it as alkylation, right? Methylation is one of the examples. There could be insertion or deletion of a base or nucleotide, whatever you want to call it. And then there could be some base analogs which are incorporated, right? So base analogs are like hypoxanthine is an analog. So it's not a normal base, but it is something else which comes up, and it can base pair with multiple, other, DNA bases, and it will basically alter the DNA. It will cause mutation, substitutions, things like that. Fine. Then coming to the two base alteration can happen due to UV induced damage. So if there is like UVB radiation, it can cause formation of something called thymine dimers. Similarly, there could be production of adenine dimer also using through the UV. Um, uh, Sorry, just give me a second. Yeah, through UV damage, it can cause that as well. So these are formation of pyrimidine dimers, which essentially stalls the replication, transcription, these kind of processes. Other things that could happen could be the chain breaks. So chain breaks could be due to ionizing radiations. So like gamma rays, alpha rays, these kind of rays could cause that kind of problems. Then the radioactive disintegration of backbone. There could be oxidative free radical formation, like the reactive oxygen species that I mentioned about, like there is superoxide radical, hydroxyl radical. These can also be reasons behind chain breaks, right? Then there could be cross linkings. So cross linkings could be between the bases in the same strand, same strand. So see, if this is the DNA structure and these are the bases which are there, so cross-linking could happen in the same strand. So let's say these two structures, they can form some cross-link, or it could happen in the opposite strand, something like this. So this kind of cross-linking damage could be there, same strand or opposite strand. So based on that, we either call it intra crosslink or inter crosslinks. And then there could be a crosslinking between the bases and protein molecule. So if we are talking about protein, here we mean histone majorly, because histone is the main protein that is involved in compacting the DNA structure, right? So these are some of the things. Then what are the factors for DNA damage? So if we can broadly categorize into the factors as the endogenous factors or the exogenous factors. Endogenous factors means these are the factors which are present within the cell, and there are some errors which happen within the cell through the machinery which is present already within the cell, and they cause damage, DNA damage. If we talk about exogenous factors, these are outside the cell and they cause DNA damage. So let's look at these factors. So, and we will talk about all of these in detail. So endogenous factors. So these are present within the cell. So the very first thing that could happen is that DNA multiplies, or it copies itself through the process called replication, right? So there could be some errors in replication that could cause DNA damage. Can everybody follow me so far? You can just give me a thumbs up if everything is clear till now. Yeah, perfect. Replication error. There could be a DNA base mismatch. So let's say cytosine is supposed to, base pair with guanine, but for some reason it's not base pairing with guanine, it's base pairing with something else. This kind of mismatches could be happening. Then there is the topoisomerase DNA complex. So topoisomerases are group of enzymes which help in uncoiling the DNA. So if you remember that DNA, whether in case of prokaryotes or in case of eukaryotes, they are in a highly compact structure. They are in a supercoiled form. Now these topoisomerases, they help in, these are a group of enzymes which help in unwinding the DNA, so that this DNA is now available, or it can, the proteins for replication and transcription, they can access this DNA for further processing. So there could be some damages, there could be some errors in this process as well. There could be spontaneous base deamination. So as I already said, a deamination process could happen within the cell, and that could also lead to some damage, right? Then abasic sites could be produced where, through the action of glycolases, which are present inside the cell, usually, they will it will cleave the glycosidic bond between the sugar and the base, and the base will be removed. And what would happen is we will now, okay, just give me a moment. We will not have the base in the DNA. There could be oxidative DNA damage. So due to various cellular processes like respiration, there is electron transport chain, there is transfer of electrons, a lot of energy is transferred during and all throughout this process, there could be production of free radicals which could cause oxidative DNA damage. Then there is DNA methylation. So these are the endogenous factors. What are the exogenous factors? Exogenous factors are like ionizing radiation, as I said earlier as well. So ionizing radiation is not usually present in the cell, but let's say if you get something like, you get an X-ray done. So if in an ideal scenario, if you do not get X-rays very often, but if it is something, if you are, if you are a personnel who performs this, then it can be a tricky situation. It could damage your DNA. Similarly, there are UV radiations, which are not usually present in cell, but again, the UV radiations could cause damage. There are alkylating agents. Then there are aromatic amines. Aromatic amines not usually present, but through cooking, there are various processes which produces aromatic amines. Similarly, there is polycyclic aromatic hydrocarbons. Then there are reactive electrophiles. These are all exogenous factors. Then there are various toxins. So people who smoke, who are, more prone to pollution, those kind of, there are some external toxins that could also damage DNA. And then there are some environmental stressors as well. There are various studies which are conducted. Uh, I mean, this is again an active field of research that your mental health and these kind of things also could alter your DNA. So anyway, uh, so talking about the endogenous DNA damage, the very first thing is replication, the damage which happens due to replication errors. So we all know that the process of replication is nothing but how DNA produces itself, right? DNA forms more DNA. So there is a replication fork. If this is the DNA, let's say, and this is the replication fork at the end of the DNA. I mean, it opens basically, and there is an enzyme which polymerizes these two strands. So if these are two strands, then they will we will have another new daughter strands being produced using an enzyme called DNA polymerase. Now, these DNA polymerases have something called fidelity. Fidelity of an DNA polymerase is how accurately that polymerase can actually, replicate the DNA. If it can, uh, if it can incorporate right bases and in the right amount, all these kind of things, this is all maintained by the DNA polymerase's fidelity. And if you remember, or if you know, or if you do not know, I'm telling you that a DNA polymerase usually has three functions. One is the 5' to 3' polymerization. Then there is the 5' to 3' exonuclease activity, and the 3' to 5' exonuclease activity. So if you know about the basics of molecular biology, DNA polymerases always work from 5' to 3' direction. This is almost always known. Uh, in case of the other strand where it has to go from 3' to 5' direction, there it goes for, forming Okazaki fragments. Only a fragment of DNA is produced. Then those breaks are joined using DNA ligase. These are some molecular biology concepts. Now, 5' to 3' polymerization is the most essential factor or element of a DNA polymerase. 5' to 3' exonuclease, what happens it removes the primers or priming sequences, extra DNA, whichever is present. So this is the main role. And 3' to 5' exonuclease, this property is also known as proofreading. So it checks whether if there is any erroneous base which has been added. In such a scenario, it will remove that base and it will incorporate the correct base. So this is the thing. Apart from DNA polymerase's own fidelity, there could be other factors as well, which could lead to, replication error. So there could be a problem in DNTP homeostasis. What does this mean? So DNTPs are the major, these are the precursor elements which are present. If you have done a PCR, DNTP mixes are added. So this, consists of ATP, TTP, and then there is CTP and GTP. These are the main, bases which are present in an optimal amount in your cell so that they can be added, right, as and when required. But if this homeostasis is disturbed, let's say there is a lack of availability of DATP, in such a scenario, maybe instead of DATP, instead of adenine, maybe thymine is added. So this kind of, disturbance in the DNA homeostasis could lead to replication errors. Right. Then there could be post-replication errors as well, which could happen due to mismatch repair pathway. So if this mismatch repair pathway doesn't function properly, that could cause, some DNA damage. Then the selection of polymerase. So as I already said, that different polymerases have different fidelity and they have different proofreading abilities. So if we are not using a good polymerase, polymerase which has low fidelity could cause more errors, right? Mm. Then there is the proofreading activity and also the thermodynamic stability of the DNA. Right? These are some of the factors. Uh, well, just let me check. Okay. Talking about replication errors, there are this could happen due to base substitution. I mean, what could happen is base substitution, or there could be insertion or deletion in the DNA. These are the three fates which could happen due to replication error. And the frequency of this error in a cell, frequency is around 10 to the power minus 6 to 10 to the power minus 8. Errors, we could say, per cell per generation. So whenever a cell is dividing, so if you remember that there is an entire cell cycle which it goes through. So in a single generation, in one cell, there could be 10 to the power minus 6 to 10 to the power minus 8 number of errors that could happen due to replication, right? This could also lead to the change in the reading frame of the protein. So whenever proteins are produced, if you remember, we talked about codons. So these are three sets of nucleotides, right? And these three sets of nucleotides, they determine what would be the amino acid that would be generated. So if there is a single base insertion or a two base insertion or deletion, any of these two, the reading frame could be shifted, and that would lead to an erroneous protein formation, right? There is something called strand slippage. I'm not sure if if you know this. So what happens is, in case of strand slippage, and one of the classic example is Huntington's disease. So you can read about it. So if there are multiple C's, it could be any nucleotide, not only C's. Like, for example, there are multiple C's or there are multiple A's in a DNA. So what happens is the DNA replication machinery, the polymerase, will incorporate thymine if it is A, let's say, then it will incorporate thymine in the opposite strand. That is how it should happen. But since it is a long stretch of DNA, it's a long stretch of DNA which has repetitive elements, let's say A, for example, so the polymerase can slip, and while slipping, and it's still incorporating thymine, it's again forming thymine. So there are some thymines here, uh, and the strand slips due to the slipping of this polymerase, and it is still continuing with the replication machinery. The replication is happening, but one of the strands has more T's than the other one. So this is how there could be insertion of bases, which can happen due to the strand slipping, and there is, looping out of one of these strands. Then there is base mismatch. So what happens is, there is, in a normal case, and when the DNA is not damaged, there is base pair complementarity. That simply means that A will, base pair with T, and C will base pair with G. This is simply what it means, right? But if there is complementarity, that is what we call base mismatch. So to correct this kind of error, there is mismatch repair proteins rather, right? And if there is any failure to detect this, for any case of DNA damage, it is very important for these DNA damaged proteins to detect this DNA damage initially. Once it is detected, then it will be repaired through various processes. But if there is some problem in this detection, or there could be some problem in the repair process itself, that would lead to mismatches happening. So regarding this, there are two main concepts. Either it could be transition or transversions. So transitions are when, a purine is replaced by another purine, then we call it as a transition, or a pyrimidine is replaced by a pyrimidine. Right? This is what we call as transitions. Transversion mutations are when purines are replaced with a pyrimidine. So just to get this, straight, what are transitions and what are transversions? So if there are like A, G, and then C and T, so if A and replaces with G or vice versa, or C replaces with T or vice versa. Those kind of in these situations, it is transition. If what happens is A is replaced with T, or G is replaced with, sorry, A is replaced with C, or G is replaced with T, in such a scenario, it's transversion. Yeah. Okay. Right. Um, Well, now talking about topoisomerases, we already talked that topoisomerases are the enzymes which are responsible for relaxing the DNA supercoils, right? These are present abundantly in both prokaryotes and eukaryotes. So it's universally present across all organisms. And these are very important to the function of this enzyme is to relax supercoiled DNA. If you know that, if let's say in case of E. coli, there's a circular DNA, but it doesn't remain circular. It's usually supercoiled like this. So these supercoils are, are unbounded using DNA topoisomerases. So what if we see TOP1, topoisomerase 1, it produces a nick. So a nick is, in, let's say this is DNA, a nick would be a small, break in one of the strand, it will cause a break, and this break will help the DNA to uncoil itself. So if you see, if you make a knot, a very complicated knot out of, let's say a thread, then it is very easy to, it's very difficult to, unwind it. So maybe if you cut one of the threads, cutting will make it easier to, remove the knot. That's how it is in case of DNA as well. So due to this production of NIC, and there is the process of, unwinding of DNA, there could be production of DNA lesions, right? And these DNA lesions are damaging. There could be formation of DNA adducts also. So adducts are when, there is some other group which is interacting with DNA, or two bases are, interacting with each other. These kind of, formation might happen due to the change in the conformation of DNA. Okay, now talking about base deamination. Base deamination is removal of amine group from a base, right? So this removal of amine group is more common in single-stranded DNA than in double-stranded DNA, right? And what kind of deamination reaction happen? If it happens with cytosine, deamination of cytosine will produce uracil. We already discussed about it. If adenine is deaminated, it will form hypoxanthine. If guanine is deaminated, it will form xanthine. If 5-methyl cytosine, and this is the most common form of deamination, 5-methyl cytosine, if it is deaminated, it will form thymine. So the most frequently deaminated bases are 5-methyl cytosine. This is the most frequent one. And the next one is cytosine to uracil. So what happens due to this deamination reaction is what we already said. The base pairing changes. So if now cytosine forms uracil or 5-methyl cytosine forms thymine, so what will happen if C is changed to T? So instead of C, which will have complementarity with G, now we will have T in that place, which will be complementary to A. So with a normal process of replication also, we will have a DNA substitution that is incorporated permanently in the cell, right? Then there is like A basic site. A basic site is are sites in DNA that lack the purine or pyrimidine naturally due to DNA damage, right? So how does this happen? So the N-glycosyl bond that is between the nitrogen base and the sugar, that is the ribose sugar, this bond is hydrolyzed using the enzyme glycolases, right? So this is the thing, basically the main concept. Coming to oxidative DNA damage. So how do this happen? So the main factor behind oxidative DNA damage is ROS, that is reactive oxygen species. How are ROS produced? ROS are naturally the byproducts of cellular respiration. Could also happen due to some, cellular respiration in case of electron transport chain in aerobic organisms. It could also happen due to some anaerobic processes, um, peroxisome metabolism, and catabolic oxidases. These are some other processes which could also form ROS. The most prominent ROS are, like reactive oxygen species, are superoxide radical O2 minus, or it is hydroxyl radical or hydrogen peroxide. All these are toxic to cells. And there are various methods, various other pathways to neutralize the action of these reactive oxygen species. So the hydroxyl reaction, radical, which is produced during the Fenton reaction. Fenton reaction, let's talk about it. So let's say there are various enzyme clusters in very basic processes like glycolysis, which have this iron, as a major element. So Fe2 plus, Fe3 plus is present. In the presence of hydrogen peroxide, it forms Fe3 plus and it generates a hydroxyl ion. This hydroxyl ion is damaging to the DNA bases. So one of the examples of this would be, like let's say guanine. This is the structure of guanine. Okay. This is taking some time. This is how guanine looks like. In the presence of a hydroxyl radical, it forms 8-oxoguanine. So what happens is very simple. This structure is 8-oxoguanine, a damage which is caused by reactive oxygen species. 8-oxoguanine can form 8-hydroxyguanine very easily, which is a reversible process actually. So if you see the structure, it's very easy to understand how that happens. Um, Instead of oxo, there will be a hydroxy group, right? And we will have something like this. Yeah, this is another thing which can happen. Another thing, this again, more amount of hydroxyl ion will cause further damage and formation of partial, uh, you know, or fragmented purine structure, which is called formamidopyrimidine. So it looks something like this. If you see, these structures are not very important. I'm just drawing this so that it's easy for you to understand how these damages happen. So one of these, rings are broken. The imidazole ring is broken, and this structure is formamidopyrimidine. This is happening due to the oxidative DNA damage, right? Another thing, another kind of damage could happen with purines as well. Sorry, with pyrimidines as well. For if we take the example of thymine, it looks like this. This is the structure of thymine. So due to hydroxyl radical, it can form thymine glycol, which is the structure is like this. So this is the structure of thymine glycol. These are all unusual bases, and these are damaging to the cell. So I hope you guys have been following till now. Then there is DNA methylation. DNA methylation is, methylation are heritable epigenetic changes that involve transfer of methyl groups. Okay, that is DNA methylation. What we mean by epigenetic is, so any kind of genetic change is a change in the DNA. For example, A is changing to T, or if there is insertion, deletion, any of these changes are genetic changes. Epigenetic changes are changes in the process, in the function basically of the DNA, but that does not involve the change in DNA itself. So let's say methylation is one of the examples where if DNA gets methylated, the further process, whatever happens, transcription, translation, they could be affected. So that is why it's called an epigenetic change. This happens usually due to DNMTs. This is DNA methyl transferases. These are the group of enzyme. The most common, change, methylation change, happens in case of cytosine. So we look at it. Instead of a H here, we will, due to the action of DNMTs, there would be a 5-methyl cytosine. The major methyl donor for methylation is S-adenosine methionine. If you remember, S-adenosine methionine is also a cofactor for type 1 restriction enzyme from the previous lectures. So this is the methyl donor, and spontaneously in a cell around 4,000, N7 methyl guanine is produced. Around 600 N3, these are the positions of where methylation happens. Methyl adenine are produced, and then around 10 to 30 O6 methyl guanine are produced. So methylation per cell per day. That is the extent of this kind of a change that happens in DNA. So methylation is a normal process. It happens in a cell normally and is not harmful ideally, but there could be several factors, exogenous factors, which could amplify the process of methylation. So these are usually N-nitroso compounds which are present in tobacco smoke, various pollutants that you might be exposed to, or choline, betaine, these kind of compounds that can accelerate DNA methylation process, right? Um, this O6 methyl methylation, this is highly damaging to the cell. Fine. Then we will talk about the exogenous damages. So exogenous damages, the first comes as ionizing radiations. These are high energy radiations, right, which can disrupt the covalent bonds. It can happen due to X-rays, gamma rays, or alpha particles. You know about these. They can cause, these are ionizing radiation, which can damage DNA. They usually cause double-stranded breaks, right? And double-stranded break is one of the most common forms of DNA damage. And it is either repaired through homologous recombination or non-homologous end joining. We will talk about it later. So due to the DNA damage which is caused by the ionizing radiation, there could be either double-stranded breaks or single-stranded breaks. So if, let's say, ionizing radiation can cause either double-stranded or single-stranded breaks, or it could also cause, because of the energy which they have, they can form reactive oxygen species in the cell, right? Free radicals are produced. Now, these free radicals will again then form double-stranded or single-stranded breaks. And the ultimate fate of this is cell death, right? So that's how it happens. If we talk about UV, UV radiation is another exogenous factor, which is the most effective and carcinogenic agent, right? It causes cancer that can damage the genome integrity of a cell. Now, due to UV, there could be various, photo products that can be formed. If we talk about UVB in specific, it can form cyclobutane pyrimidine dimers. These are CPDs, or they could form pyrimidine 6,4 pyrimidine photoproducts. These are also called 6,4-PP, and they can further form Dewar isomers. These are some damages which can happen due to UVB radiation in specific. Thymine dimers could be formed. We already discussed about it. In the same, strand, two thymines together, stacked together, they can form dimers. They can also form adenine-adenine dimers. So CPDs, these all are cytotoxic to the cell and they are damaging to the cell. Well, after UV radiation, there are alkylating agents. So alkylating agents, they add alkyl group, right, to the bases. That's the function. Now, alkylation can happen at the N residue or the O residue. Okay. O alkylation is more toxic than N-alkylation. N-alkylation is more common, likewise. And these kind of alkylation is repaired using base excision repair, right? Uh, right. Okay. Another thing which could happen is aromatic amines. Aromatic amines are basically, these are organic compounds with amine ring. Sorry, organic compounds which have a ring which has an amine group added to it, right? So this is what aromatic amines are. So there are heterocyclic aromatic amines which are produced when you fry, meat products. So through protein pyrolysis, they can form heterocyclic aromatic amines which can cause cancer, which have the DNA damaging property. So this is all about aromatic amines. Coming to DNA adducts. DNA adducts are DNA with some other chemical group. If it is attached to DNA, it is called a DNA adduct. So it can also interfere with replication or transcription and can induce mutations, right? These are the implications. Then we have cross-linking. What happens in cross-linking? We already said it could be of two types, either intra-strand, where within the same strand there is cross-linking. That kind of cross-linking is called intra-strand. It could be inter-strand, where two different, bases, they cross-link. That is in inter-strand. They also have cytotoxic effects, so can cause cell death, right? So due to this DNA damage, whatever we have studied, all this DNA damage can lead to various diseases. One of the most common one is cancer, right? Cancer being the most common one. And in case of cancer, it's not usually one particular mutation that leads to cancer. There is accumulation of mutations, accumulation of DNA damage, which is beyond repair, which cannot be repaired anymore. And that's why it becomes a problem. That's why how cell becomes cancerous. The properties of the cells also change, right? In case of a cancer. Apart from cancer, there are neurodegenerative disease like Alzheimer's, Parkinson's, which are also, linked to DNA damages, and even diseases like COPD, that is chronic obstructive pulmonary disease. You must have heard about it in case of COVID. During COVID, there were several morbidity cases like CVD, COPD, where patients were more susceptible to die with a COVID infection. So if a patient is already having a pulmonary disease, a lung disease, that patient is more likely to die if he or she has COVID, right? So yeah. Then we will discuss about the DNA damage pathways. DNA damage repair pathways. So one of them is photoreactivation. Photoreactivation, then there is base excision repair, nucleotide excision repair, mismatch repair. There is something called translesion synthesis. Then intrastrand cross-link repair. There could be single strand break repair or double strand break repair pathways, right? Let's talk about each of them very quickly. In case of photoreactivation, what happens is the damage which happens usually due to UV rays can be corrected using an enzyme called photolyase. We'll talk about this enzyme very soon. What happens is, let's say due to UV in a DNA, if a UV ray falls on it, most commonly UVB, it could cause some damage, let's say in this part of the DNA, right? It causes some damage. And one of the most common reasons could be, CPDs. Now, what happens in this DNA? An enzyme called photolyase, it will come and it will, this enzyme will bind to this damaged part and it will essentially correct this damage essentially in the end. So this will be corrected. So this is the process of photoreactivation. For photoreactivation, the enzyme is DNA photolyase, which is a monomeric flavin-dependent enzyme. Right? It has around 420 to 616 amino acids, and it has two cofactors. One of them is the catalytic cofactor. Another one is the light harvesting cofactor. The catalytic cofactor is FADH, reduced FADH, which is an electron donor. And the light harvesting cofactors are MTHF or 5,10 methynyl tetrahydrofolate and 8-HDF. There could be MCQs which could come up from here. What is the importance of light harvesting cofactor? It absorbs light and it transfers the energy to FADH. So also, one another important very factor for photoreactivation, which I forgot to mention, is the importance of light, near UV light, which is near to the visible spectrum. Right? Fine. Let's go to the next slide. Base excision repair. This happens in the G1 phase of the cell cycle. Most commonly in the G1 phase of the cell cycle. It happens through enzyme called DNA glycolases. Right? If you remember, DNA glycolases are also the enzymes which are responsible for creation of A-basic sites. So if there is an incomplete base excision repair pathway, it could lead to generation of A-basic sites. So now these DNA glycolases could be monofunctional. Monofunctional meaning they only have the glycolase activity, or they could be bifunctional. So they have both glycolase and beta lyase activity. So this base excision repair can be, there are two pathways for base excision repair. One is the short patch pathway, where monofunctional glycolases, they go for short patch pathway. Bifunctional glycolases, they initiate the long patch pathway. You can read about these pathways in the lectures, but this is just for the concept of it we are discussing. And then we are coming to nucleotide excision repair, NER. Nucleotide excision repair is usually for bulky lesions. That means if there are, there's an excessive damage which happens, in that case, we go for nucleotide excision repair, right? Now, nucleotide excision repair can also be of two types. It could be global nucleotide excision repair, or it could be a local, or what we call as transcription coupled nucleotide excision repair. So how are they different? In case of a global nucleotide excision repair, it can happen anywhere in the genome, right? In case of transcription coupled nucleotide excision repair, it is an accelerated nucleotide excision repair for the transcribed genome. Essentially, the part of the genome which is transcribed, which forms an RNA, only those elements or those parts are accelerated in terms of repair pathway. There are various proteins which are involved behind it. Please read them through the lectures, right? After nucleotide excision repair, there is mismatch repair. Mismatch. Let me see. My iPad is. Yeah, it's fine now. Mismatch repair, it is good for short insertion or deletion, which might happen in repetitive DNA sequences. Right? And this, if you remember, we said that in repetitive DNA sequences, there is something called strand slippage, right? So if there are strand slippage events and there is, short insertion and deletion, that could lead to mismatch repair pathway getting activated. It is also highly conserved in eukaryote and prokaryotes, meaning this is a very important process for DNA repair, right? And mismatch repair increases the replication fidelity by 100 folds. So if mismatch repair pathway is not functioning correctly, that will lead to various problems, various DNA damages, right? If we talk about the steps, so the first step of any kind of repair pathway, we already discussed about it, is the detection step. So detection of a mismatch site is usually done by MUTS or its homologue. This identifies this mismatch site, and then this sensor, it activates a set of protein downstream. So that nick this DNA strand and remove the mismatch base. After this, what happens? A DNA polymerase comes in and it resynthesizes the damaged strand. After which there is DNA ligase, which will finally join the strand. So this is how the process works. There are also multiple proteins which are involved behind mismatch repair. You can read about it. Then there is something called translation synthesis. So whenever in a DNA, let's say this is a replication fork, right? And if there in a DNA there is a lesion which will prevent the replication or transcription, to happen further because it's a lesion, uh, there could be a process of repair which is called translesion synthesis. Now, this repair can be processed through two pathways, either polymerase switching or there could be gap filling. So what happens in case of polymerase switching? And these polymerases involved are highly conserved TLS polymerases. We'll talk about it. So what happens in case of polymerase switching? So let's say in this case, if there is polymerase switching, this is the replication fork. The polymerase will go further in one of the strands, which is undamaged strands. And here there would be another polymerase, which is the TLS polymerase, which will come and repair in case of polymerase switching, right? In a gap filling model, what happens is, so through replication, there are two strands. One of the strands is damaged, the other one is normal. So the other one will be, going about its normal process. But the one which is damaged will have some breaks inside, right? Some gap, essentially. And this gap is filled through polymerization, right? That is another gap filling model, and it is the exact mechanisms are not very clearly known. Then there is intrastrand crosslink repair pathway, where two bases from complementary strands are interlinked like this. So if this is how they should be, linked in across the same stack, but if it is happening, in some this way, then it is interstrand crosslinks. Now, these are highly toxic lesions because they can stall replication and transcription, because any enzyme which will try to work with this kind of DNA in these sites will not be able to, go for a normal replication cycle. So there are some Fanconi anemia proteins, FA proteins, which are cell cycle dependent, proteins. So essentially, this kind of damage is repaired during the G0, G1 phase or the S phase of the cell cycle. Fine. Uh, then we have the single-stranded break repair pathway. So here also there is the long patch pathway, which involves identification of the damage using PARP1 and the end processing using enzymes APE1, PNKP, and aprataxin. In case of short patch pathway, identification is through APE1, and then there is gap filling and ligation. Fine. Similarly, for double-stranded DNA breaks, so this is very simple. If this is the double strand, these DNAs are broken like this. Now, there are two ways by which this kind of breaks can be repaired. One is the non-homologous end joining, where essentially these two breaks are simply joined together using the help of some proteins called the Ku proteins or Ku proteins. In case of homologous recombination, what happens, there is another set of DNA, which it is a more precise repair pathway, where there is a template DNA which is present. And then there is formation of a structure called a holiday junction. If you look at the entire process, you will understand. So here what happens is there is recombination of this two strands. So it will look something like this. So there is a structure called holiday junction. So essentially there is recombination of, I've drawn it wrong, I'm really sorry. it should be from here, and from here it should go here. So there is a holiday junction formation. Based on these repair pathways, there are various diseases which could happen. So one being the Fanconi anemia, then xeroderma pigmentosum. You can read about which particular pathway is involved behind these diseases: cocaine syndrome and trichothiodystrophy. Dystrophy. So let's discuss about questions now. The first question is, transversion is any mutation where a purine base is replaced with a pyrimidine, or a pyrimidine is replaced with a purine. B, a purine base is replaced with another purine. C, a pyrimidine is replaced with another pyrimidine. Or D, a purine or pyrimidine base is deleted. You can use the Q&A section to answer. Let's give this a minute. Not receiving answers yet. Transversion is a mutation where which of these following four happens?
So the answer would be
Option
option
A.
A.
Right, correct. The answer will be option A. A purine is replaced with a pyrimidine, or a pyrimidine is replaced with a purine. You can give your answers in the Q&A side. It's easy that way to follow. Otherwise, you can also give answers like this. The key function of DNA topoisomerase is to replicate DNA, to synthesize mRNA, to synthesize protein, or to relax superhelical DNA. Okay, now I'm getting answers. So the answer is D, to relax superhelical DNA. We use DNA topoisomerases. Question number 3. Deamination of adenine produces A, hypoxanthine, B, xanthine, C, uracil, D, thymine. You can use the Q&A for answering. Okay, A, hypoxanthine. Yes, that is the correct answer. Which of the below is an endogenous DNA damaging factor? Is it A, replication error, B, ionizing radiation, C, UV radiation, or D, environmental stresses? Okay, getting answers. Okay, A is the answer to this, as already pointed out. Question 5, photoreactivation repairs UV-induced DNA damage, or B, replication error, C, interstrand crosslinks, or D, A-basic sites? You can answer in the Q&A site, or you can raise your hands as well if you want to answer. It's again a UV-induced DNA damage. Bulky DNA lesions such as cyclobutane pyrimidine dimers and 6,4 photoproducts are repaired by base excision repair, mismatch repair, nucleotide excision repair, or photoreactivation. What would be the answer to this? You can raise your hands or write in the Q&A.
Sir, the answer to this question would be photoreactivation.
Well, photoreactivation can actually repair CPDs, cyclobutane pyrimidine dimers, but not essentially 6,4 photo products. So if the lesions are bulky, the answer should be nucleotide excision repair. So if you remember, when we talked about nucleotide excision repair, we talked that bulky lesions, if they happen in DNA, they are repaired through nucleotide excision repair pathway. So yeah. Question number 7. Xeroderma pigmentosum is a rare autosomal recessive genodermatosis caused by mutation in which of the following? DNA replication, nucleotide excision repair, transcription, or base excision repair. So if you have read about these, well, you can answer. Any answer? Anyone would like to answer? It is B, yes, nucleotide excision repair. Coming to question 8, in DNA photolyase, reduced flavin adenine dinucleotide FADH works as a catalytic cofactor or a light harvesting cofactor, metal ion, or an antenna molecule. What would be the answer? This is an interactive session. It will be more interesting if everybody joins in. They give answers either in Q&A or through you can like unmute yourself and just answer. Anyone would like to answer? Nobody. Catalytic cofactor is FADH. If you remember, this is the answer. Coming to question 9. Topoisomerase mediated DNA damage. TOP2 binds to dash double-stranded DNA at TOP2 recognition site. Is it A, G2, GT, B, CG, CAT, or DTU? Any answers? Not getting answers. Only one of you are responding in the Q&A. What about others? Okay. So the answer to this would be GT. Which of the following is a DNA repair mechanism? Is it replication error, intra or inter-strand crosslink, photoreactivation, or all of the above? Which of the following is a DNA repair mechanism? We talked about it in detail.
Option C, sir.
Yes, C. C is the right answer. Thank you. Question 11. Which of the following proteins play key roles in the rapid global single strand break pathway? Is it A, PARP1 and XRCC1, B, DNA polymerase and DNA ligase, mismatch repair proteins, or telomerase and helicase? I think it's very easy.
Option A.
Right, correct. Which component of solar radiation is responsible for bringing about chemical modifications in DNA and altering its molecular structure through the formation of dimers? Is it A, infrared radiation, B, visible light radiation, C, UVB radiation, or D, X-ray radiation?
It would be UVB
Option
radiation.
C.
That is absolutely right. That's why we use sunscreens, right? So yeah, the initial step of mutagenic pathway of aromatic amines, which involve aromatic amine ARNH2, involving N-hydroxylation. Which enzyme is involved? Is it aryl hydroxyl amines? Is it guanine nuclease, cytochrome P450 enzyme, or DNA nucleobases? Well, it's an interactive session. Please interact. Can all of you not access the Q&A's part? Not getting answers anymore.
Sir, could you please help it? I cannot recall the answer for this one.
Well, in that case, if you remember, in those lectures, aromatic amines are converted to, if you remember the mutagenic pathway in case of aromatic amines, it gets converted to uh, hydroxylated amine, aromatic amine, using an enzyme. If you remember, it was CYP1A2. What is this enzyme? That is the question that they are asking. After this, there could be three fates. It could be N-acetyl transferase, one enzyme, or O-acetyl transferase, or sulfotransferase. They can lead to, N-acetylated, aromatic like hydrocarbon, or O-acetylated aromatic hydrocarbon, or uh, sulfonated hydrocarbons. These are some things which can be formed, and they can further be reduced to form nitrinium ions, which are carcinogenic. But this enzyme is a cytochrome P450 enzyme. It's given in the lecture slides as well. Is it clear?
Yes, sir, it's clear now. Thank you.
Question 15, in E. coli MMR mismatch repair pathway, which protein specifically incises the unmethylated DNA, nascent DNA strand of hemimethylated DGATC, serving as the initiation site for mismatch provoked excision? Is it MUTS, MUTS, MUTL, MUTH, or DNA helicase 2? E. coli MMR pathway has been it has been described in detail in the lectures. Anyone would like to answer? Okay, so the answer is MUTH, right? Which of the following statement is true for Fanconi anemia? Fanconi anemia is a dominant, just give me a moment. It's a dominant genetic disorder characterized by chromosomal instability. Uh, Individuals with Fanconi anemia have a decreased risk of developing acute myeloid leukemia. Mutations in only one gene have been linked to the development of FA, Fanconi anemia, or Fanconi anemia is associated with DNA damage and repair and interactions with proteins like ATM, BRCA1, and BRCA2. If you read about this in detail, whatever is produced in the lecture slide, you can find the answer. Anyone would like to answer?
Option D.
That's correct. Absolutely correct. Question number 17. Base excision repair is mostly active in which phase of the cell cycle? G0, G1, G2, or S? I think we talked about it today itself.
G1.
One
Right.
phase.
Not G0, it would be G1. G0 and G2 phase, G0, G1 was for, I think, translation synthesis and S phase as well. For base excision repair, these are the answers. In a transition type DNA mismatch, in transitions basically, leading to DNA damage, which of the following is responsible? Purine is changed to another purine, or purine is replaced with a pyrimidine, or a pyrimidine is replaced with a purine, or a radioactive disintegration of the backbone element. What would be the answer? I think it's very easy.
Purine nucleotide changed into another purine.
Absolutely correct. Okay. Question 19, and I think this is the last one. What can cause a change in DNA reading frame leading to insertion and deletion of nucleotides? Exposure to visible light, base excision repair, slip strand mispairing events at repetitive sequences, or DNA polymerase error during replication?
C, sir.
Option C.
You're right. Okay. Well, that's how we are ending this session today. If there are any questions from your side, now is the time to ask. Any questions? Okay, any feedback from your side? If you would like to, tell me if I'm going too fast, too slow, nothing is understandable from your end. If those kind of feedbacks you can also give. Anything anybody would like to say? How is the course going for all of you? Is it all fine?
Yes, sir, it's fine. It's just
Good.
it takes us some time to understand things or learn them, but yeah, like slowly and gradually we'll be able to do it.
This is a very interesting course. A basic understanding of molecular biology is required to grasp some of the concepts. So I had this course, this very course in my MTech in IIT Guwahati. So I did my MTech from there. So it's a it's a good course overall. It will be helping you pretty well. So if there is nothing, then you guys can leave. If anybody would like to talk about anything, you want to ask about anything, or you want to introduce yourself, maybe if you were not present in the last session, you can do that. Otherwise, we can just call it a day.
Thank you,
Yes,
sir.
Rutuj. Okay,
Yeah.
thank you.
No, I'll just leave it, sir. Thank you.
Anything from your side, Krishnendu or Shushmita?
No, sir. Thank you, sir.
Okay.